let's discuss genes and proteins. So what codes for a protein? A gene found on the DNA molecule. The order of those A's, T's, C's, and G's on DNA is what makes up a gene. Those adenine, thymines, guanines, and cytosines, those nitrogenous bases, is what makes up a gene. So where do we find these genes? Well, where do you find DNA? It's in the nucleus of the cell. So almost every cell in your body has the same DNA. What makes those cells different is that only some genes are turned on and all the other genes are turned off inside that cell. The genes that are turned on, they make proteins. And it's those proteins that's what making your cells different. It makes your cells have a specialized function. So what do proteins do? Well, they provide structure and they do most of the work inside the cell. An analogy is, think of proteins as a chain. And that long chain has many different links, and those different links would be like the amino acids. So amino acids, they're the building blocks of a protein, just like each link is the building block to make an entire chain. So, as you can tell over here on the left, links in a chain is similar to the idea of a sequence of amino acids. The order of amino acids. One amino acid is attached to the next amino acid. Just like one link in a chain is attached to the next link. An analogy for protein synthesis is that DNA is like a cookbook. So when you open up a cookbook, you notice there's lots of different recipes. And if you just pick one recipe, you can think of that as like the RNA molecule. And of course, that recipe has the instructions to make dinner or to make a meal. So if you go and make one meal, you can think of that as the protein. Now, protein synthesis happens in two phases. The first one is transcription, where DNA code is converted into RNA code. And the second phase is called translation, where RNA is used as a blueprint to build a protein. Below is a model of protein synthesis. You notice we have on the left DNA, and that's double-stranded. We have our A's, T's, C's, and G's. And if we transcribe one side, taking DNA code and making RNA code out of it, and notice that RNA is a single helix compared to the DNA, which was a double helix. And if we take that RNA code and we translate that, we can build a protein, which are made out of amino acids. Transcription is going from DNA code to RNA code. But translation is taking that RNA code and using that information to build a protein out of amino acids. I hope this helps. Thanks for listening.